Hello, good afternoon, everybody. This
Yeah.
is Luciano Lucas here. I'm the exec vice president here at DR Vision, and together with me, I have Tze-Yuan Cheng, is one of the algorithm engineers at our company, and he's going to help me present today's webinar.
Hello, everyone. Good to see you all here.
So Tze-Yuan actually has done his PhD at Johns Hopkins University, and his background is really relevant for this presentation. That's why he's joining me, and he's also the guy who has been working on the classifier that we have in Avia, and so I'm very happy to have him with us for this presentation. Also helped us today organize everything. Hoyen, who is our product manager, is taking questions and looking at the live stream. We also have Queen Tran, who is also monitoring the questions in the live stream, and Willie doing our technical support and application support, who is also helping us run the whole event. So there's a few people here, just the two of us on camera, but everybody else is here to help as well. I'd like to say also, you can ask any questions you want via YouTube directly. You can also ask them by our DRV Tech Twitter account, on LinkedIn, or directly on our website. So before I move forward, a little introduction to the topic, today's topic. So we're going to be talking about machine learning, and we're trying to make it a gentle introduction for biologists. Machine learning is actually one of the main expertise here at DR Vision. We've been active in this field for over 20 years, have a number of patents in this area, and a number of applications developed for our own products and for other companies' products. And today we're talking about the very narrow application of this type of technology, but I think it's a very good introduction for the microscopy and the biology life science market. So I wanted to frame this in terms of a problem, and this is something that I felt a lot when I was doing my work and my research work, is that I often felt like there were just too many parameters and I didn't know which ones to pick. And so I tried to frame it into three questions, and the questions I'm trying to ask you is did you ever feel like you had too many parameters and you didn't know really which ones to pick to identify effect versus no effect in your experiments? Another struggle or question that I had is if you ever had difficulty to prove or disprove an hypothesis based on a limited set of parameters. And that's often needs to be the case, because it's very difficult actually to handle all of the parameters that your experimental setup can generate. And finally, being overwhelmed with the complexity of the data, not just the data parameters, but the relationships between the parameters. This is something that affects most biologists. Even with the recent advance in new technologies that help manage large amounts of data, it's still very challenging to actually get a good grip on all the complexities and make good use of that information. So most people would say, okay, the problem is we just have too much data. And this cover from The Economist made the term very popular, the term of data deluge. And that's from 2010, this cover. And it talks about all sorts of volume data in financial markets, in biology, in biotech. And it talks about it as if it is a big problem. And in fact, it is. It also creates some opportunities, and everybody knows the success that Google and Facebook and other companies are having because they have our data. But I ask, is this the problem that we are facing here as well? And so I started to look at the scale of the problem and try to see where we fit. We meaning life science group of people and the work that we do. So as a group, meaning humanity, we create about 2.5 exabytes of data every day. And that's an enormous amount of data. And that's the whole world in one day creates 2.5 exabytes of data. Facebook alone creates 500 terabytes of data, which is a relatively small amount of data. So it's only 0.005 exabytes, not that much. And if we look at a more close to home application, so looking at a zebrafish brain over 40 minutes, and if you do that 10 times in one day, you're creating very, very small amount of data. It's actually only 2 terabytes or so. So it's 0.000002 exabytes, which is a large amount of data if you consider it's just one zebrafish one day. But on the grand scheme of things, it's not that bad. It's a small amount.
So Luciano, I have
Yeah.
a question.
Yeah.
So what is EB beta?
Yeah, the exabyte. So 1 exabyte is 1 million terabytes. And
okay, 1 million terabytes.
yeah, which is about 1 billion gigabytes. Yeah, that's what is it. That's good to clarify because we don't normally use this terminology, exabyte, terabyte, and gigabyte. So exabytes is 1 million terabytes, and what we see here is that for our little space, we're not generating that much data in one go. However, if we look at the core facility, with 4 microscopes, with 30% usage load, we're looking at about the same amount of data per year as Facebook creates in a day. So again, it's not that much. So we're talking about something like 500 terabytes of data per year for one microscopy facility, which is similar to what Facebook does in one day. So again, it doesn't look like a lot of data. However, if you look at the whole community, then it becomes really a bigger problem. Look at this. It's 3,600 terabytes globally per day. So that is more than actually what Facebook is generating. So in terms of volume, this is quite impressive. And there are a few good tools to manage this amount of data, Omero and Bisq being one of them. But this is just part of the problem. This is just the volume side of the problem. But where the information really is in the depth of the data that we are gathering. And bio-related data, so in pharma, biotechs, and in research labs, we have a lot of information, a lot of depth in that data. And that's what we try to make use of using machine learning. It's not just the volume itself. So if we take 100,000 images, which is what a modern microscope can now generate, 100,000 images in one day, images that are 3 channels, each image, we're creating about 1.2 terabytes of data. If we do a 2-week experiment, that's 12 terabytes. And if we look at around 30 parameters per channel, that's 100 or so parameters, which gives us 100 million data points. Now we could look at it this way, the 2 ways. Either it's a huge problem, how do we handle this, how do we extract meaning from this, or we can turn it on its head, and that's essentially what we are doing with machine learning, is making that data work for us in an intelligent way. So it's not infrequent to feel like this, especially for more junior researchers that have maybe not yet good connections with bioinformaticians or biostatisticians or data scientists. It can often feel like you have a lot of data in volume, you have a lot of data in terms of complexity, and there's just no way we can extract all the good information out of this. It's very hard. So we use our bias. We select a few parameters that we know should be changing in our model, and we focus on those. And essentially everything else is shelved, is not used, and it becomes kind of wasted, or at least stays on standby until another question comes along. So we're trying to change that a little bit. We're trying to, together with the wider community, make that problem an opportunity, convert it, and making good use of the data depth and the data complexity is something that machine learning is very, very good at. And we're trying to bring that kind of power to a broader audience, to everybody here listening today. And we have researchers, but also some industry partners present, and so we want to make that available to the bioimaging community in the best possible way. So we believe it's certainly part of the solution. It's not a big problem, it's manageable, and we can actually create good solutions for it. So this image here represents, let's say, an experiment with lots of data points, 100,000 as we had, 100 million as we had on the previous slide. And there are data points and there are connections between the data points. And the data points themselves have value, and the connections also have a value. So we want to keep all of that information and not lose it. So we take that data, we use the expertise from a knowledgeable scientist, so whoever is the person running the experiment. In this case, the demo that we're doing today is for neurosciences. So a neuroscientist would look at some sampled images and manually assigns classes to them. Once that is done, the system is essentially trained, and we can start to gather meaning from those images in a very automated way. So in a way, this is a simple artificial intelligence deployment that we're creating here, a simple AI that we're generating using the models that we're presenting today.
I see.
So the workflow that we have goes from data pre-processing all the way to the classification or to the prediction of classification. So we take lots of images, we segment them. In our case, they are neurons, or the demo that we have today, there are neurons. We have then lots of parameters, and we can also extract lots of features from these images. We use the features to train because we know, or the user will be able to define that this is one class. So this cell, and this cell are of one class, and the other three are of a different class. And by doing that, it's informing the model that we have, the machine learning model, that cells with that set of features are of a particular class. Once we have built a classifier, so that's the training process, we teach the classifier. This classifier can then be applied on any number of data sets automatically, and very, very quickly will discern between the different classes, and will group all the objects that are detected into the classes that we have pre-trained. Or you can create your own classifier.
I have another question.
Yeah.
So you mentioned the parameter and features.
Yeah.
So may I know what's the difference between them?
Yeah. So a parameter is more, it's closer to what the biologist would recognize.
I see.
So a parameter would be cell size, so diameter or volume or intensity. So those are all related to real measurements that the biologist would normally do either by eye or with some segmentation software. A feature, however, is we can have many more times the number of features as we have of parameters. So we may have 30 parameters only calculated, but each parameter can have multiple features. So for example, if you look at intensity, we take nuclear intensity like we have here, we could look at relationships between adjacent pixels, and that would be another feature. So what is the intensity ratio between this pixel and the pixel right next to it? That would be a feature which is related to intensity. We can also look at the distribution of intensity, and those are texture related features. And so we can have easily 3 or 4 or 10 times more features than we have parameters. and we can use all of those features to create the models.
So I think feature can be derived from the parameter. Right?
Yes, exactly. Yeah. So the feature is derived from those parameters.
Thank you.
Yeah. There are already a number of applications for machine learning algorithms. One of the earlier ones in life sciences is protein structure prediction, and that's now more than 20 years ago that this kind of work was done, and gradually more applications came along. So high content screening is a very popular one, drug development, DNA sequence analysis, and proteomics. But the list is expanding very, very quickly. The academic world, the community, has really embraced the type of algorithms that are available and tailored them for particular applications. And the pace of publications coming out that use some sort of machine learning is really accelerating. And that's very encouraging, because we can, of course, accelerate discovery and get to better therapies for a lot of diseases. So if we go now a little bit more in detail into the different types of algorithm and which ones we are using, we can start here. And I should say, in the recent years, there's a lot of talk about machine learning and deep learning, and you probably heard about these terms being used broadly. Why are they so popular now? Most of these technologies are not new. They are 20 years old, sometimes even older, 30 years old, but they are making a comeback. And why is that? Well, one main reason, especially for deep learning, the main reason is that the computing power is much, much higher at a low cost. And so that makes it possible to train rather complex models using a relatively cheap computer. You don't need to build a multi-million-dollar computer in order to be able to train a relatively simple model. Nowadays, and we have computers here at DR Vision that we build ourselves and are not that expensive, but it allows us to achieve good results already. Of course, for scaling, it makes sense to go and use distributed computing and cloud computing and so on. Now, machine learning is actually the discipline. Deep learning is just one of the sub-disciplines of machine learning. And if we try to categorize them, they essentially all have something in common, which is the training and the applying of the generalization to a bigger group of images. And if we go a little bit forward, we have two types. One type, which is supervised machine learning, which is the more popular one nowadays. And in there, in the supervised machine learning, we have a few types. One is pixel classification. That's when the user goes directly into the image and draws areas that are basically could be, for example, background, and then another area that is a cell type, and another area that is a different cell type. So you create 3 classes of pixels. And from that, you have a mask, which you can then segment on and create regions of interest or segmented objects that are of 3 different classes. That's the pixel classification. Another one, and this is useful when it's hard to segment the objects. In our case, we actually have managed to segment the objects very well using Avia to detect the 3D structure of neurons. But the next class, or the next type, is object classification, where you take the images, you analyze them, so segment them using some algorithm to detect what is neuron, what is not neuron. And once you have all of those neurons detected, you can then assign classes to some neurons, build a classifier, and then you can deploy that classifier on new images. And when it's totally impossible to do segmentation on the images, what you normally do is look at a whole image and say, on this image, most of the cells are of class A, and you assign the whole image as one class. And then you look at another image and you assign the second image as a different class, or maybe the same class. And by doing that, you teach on the whole image, not on a specific object. This can work, but in my experience, it's a little harder to make work and less informative than the object classification, because you don't get per cell information. You just have a whole image classification, which for some applications is exactly what you need. So it's, you need to find what works best for your application. In our case, we're using object classification. This type is particularly good for classifying known phenotypes, and it is populated by a number of efficient algorithms. Random forest is the one we're going to be describing today, but there are others. Adaptive boosting, support vector machines, and even deep learning could fit into this category as well. There are supervised and unsupervised versions of deep learning. We find that supervised or partly supervised deep learning is the most efficient and most powerful. On the other side, there are the unsupervised methods. These are good for finding unknown phenotypes, but are a lot harder to make to reach a good result. One such method is clustering, but it is very abstract. You have very little control over how the clustering is taking place, because it's unbiased on one hand, which is good. On the other side, you might end up with something you don't recognize. So for biology applications, it tends to be supervised and either pixel classification or object classification. Now, if we look at a little bit more detail on the supervised machine learning approach, you can see the linear decision type of creating a separation between the classes, which has high bias and low variance. However, it's very robust to noisy data, and you can see here this is not particularly noisy. There are two groups almost by eye, you can tell. There's one group on the top, one group on the bottom, but there's a little bit of overlap. Like what you see, there's one dot down here that belongs to the other group. That would be considered noise. And if you had more dots down here that are from the top group, that would be noise. And if you have a linear decision-making process, it will just draw a line between the two groups, and whatever is in the top will be of class 1, whatever is on the bottom is class 2, or vice versa. So very robust to noise, however, not so good at separating nuances and complex data sets. The nonlinear approach is what we're using in our approach, and it's much better at detecting complex, subtle patterns. It has low bias, high variance, and it's very good for complex data sets, like in biology. And it is much more sensitive to training noise. So you can see here what I mean is that it tries to match as well as possible a little bubble around the points of interest. And if you go to higher granularity, it will separate them even further, and it will try to fish this one point out of that group if you train it more or with better data. So it's very good at distinguishing subtle patterns and complex patterns. Then we have the generative approaches. So this one is discriminating approach. Then we have generative approaches, which will actually create a simulation of the morphology. And that's something that we're not focusing on today. Maybe it's a topic for a different presentation. So with that, that's the first part. I will take a pause and I'll bring back Tze-Yuan. He's been patiently waiting, and he's going to give you a little bit more background and details on our implementation, how it works, and also how we have built our classifiers.
Yeah, it's very good to see everyone here. Today I'm going to talk about some concept and some implementation about random forest algorithm, which is also the core algorithm you use for the neural classification in Avia 5. So before talking about the random forest, I think I would like to introduce what's a decision tree, which is a computing unit for random forest. So decision tree is a type of supervised learning. The goal is to construct a binary tree to classify data point based on key features. So here I'm going to introduce a very simple example in our real life. So suppose now we are a bank and we are getting a request from customer trying to approve a credit or get a loan. So we can just describe our customer as a data point with some features like age, education, and income. So, and then we can build a decision tree, and then at each node, we will try to check the condition for some key features. Like, okay, if the first feature we can check is whether the data point has age greater than 40, and then we can split into subgroup like yes and no. And in the yes group, we can further split into if the customer is a homeowner, or if it is yes, then it is potentially more reliable to be a good customer, then we'll make a decision to give a credit. But if if it doesn't satisfy this condition, we will go to another path, which is another subgroup. And if unfortunately the customer doesn't have income greater than 5,000, then will lead to a classification as to refuse a credit. So this is basically how the decision tree is working. It tries to split the data point into subgroup and then eventually make a decision at the leaf node. So then the question is, how do we get this kind of decision tree? So then I'm going to talk about the decision tree learning. So decision tree learning is actually a process to generate the feature node that best split the training data into subgroups. So for example, here if we have a data with class A and class B, and each of the data has a feature like S1 and S2 and distributed in this two-dimensional space, distributed like this. Then because decision tree is a supervised learning, so we have to make those data being labeled. Then we can try to find a good feature to make our first split. So now we can see, for example, maybe S1 equal to 1 can basically extract some of class A points totally away from the other of the data. So then we will have our first node, which is condition S1 greater than 1, then split into yes and no subgroup. So in the no subgroup, then we have a very good homogeneous distribution that there's only class B data, so we can stop splitting and make it a leaf node. But on the other side of the plan, the data are still mixed together, so we need to further split it. So we draw our second line, which is S2 equals 2, then split into the top region and bottom region. Then we can apply splitting in the same method and keep splitting into two more subgroups on the top and bottom. So eventually will lead to a subgroup in which it's very clear towards certain type. So then we will try to make majority voting at each leaf node, then to decide which class each leaf node should correspond to. So now we have a constructed decision tree using this state labeled training data. So now if we have a sample with unknown type or classes, we can run through a decision tree. So now it is in the condition like S1 greater than 1, S2 not greater than 2, but S1 is in this subregion also not greater than 3, then we'll false onto this leaf. Then we can make classification. Okay, this data is actually class B because in this leaf node the majority is class B. So I hope you have a basic understanding about the decision tree. So now we are about to move forward to talk about what is random forest. So random forest, I think by its name we can just interpret as a multiple tall decision tree. So we need to create a forest, which is aggregation of multiple decision tree. Then the next question, why we call it random? Because this, in the random forest algorithm, each tree is actually being taught uniquely because we apply bootstrapping. What is bootstrapping? Which means when we teach each individual decision tree, we'll take a random subset of training sample. In addition, when we are generating those splitting nodes, every time we will also take a random subset of features. So random forest is random in these two aspects. So in this way, we can create multiple decision tree, and each of them are actually trained differently. So so this is the process how the random forest is being generated. Then once we have a random forest, we will also do the majority vote. But this time, the majority vote is based on each individual tree. So then we will run the features for each individual tree, then try to get what's the majority for the classification that was given by each individual tree, then finally determine the sample we want to classify. So here's just a flow chart summarize the whole process of random forest training. First, we have a whole data set with labels. Then we will take a subset of data point to teach a single tree. Then each single tree will take further a random subset feature to generate the nodes of each tree. Then we repeat until we have a forest that has enough number of decision trees. So in this way, we'll create a multiple tree with different structures, and hopefully we can use this the majority voting to make our classification with minimized error. So here is a visualization of how the random forest used in training and testing phase. So in the training phase, this is the whole data distribution, data points. Then every time we will take a subsample, like subset of this training set to generate each decision tree. Then eventually we will end up with n number of trees. Then in the testing phase, each tree will give a vote for the same data point we want to classify. Then each tree might have different classification, but overall at the end we'll take the majority vote to decide what's the real class of this given example.
So let me interrupt you. We're talking a lot about random forests. And I mentioned earlier on there's a few other possibilities that we could have used.
Yeah.
Often people want to know why did we pick a random forest? Why not something else?
Yeah, that's a very good question. I think remember in your slide you talk about that nonlinear classification is more robust for complex problems, complex data set. So the random forest is actually in that kind of category. And maybe you talk about it might have high variance, but because that's for decision tree. But when we are using random forest, we actually try to minimize that variance that might possibly generate some errors. So random forest doing the majority vote to minimize that side effect. So because I think our neural morphology may look very complex in different type of different diseases. So random forest is that's the reason we pick random forest to develop our classification tool. Okay. Yeah, so now I'll move on with some key parameters for random forest learning. So in random forest, we will usually, when we are practicing the learning, we will usually need to specify what's the maximum number of trees, which is usually served as a termination condition where we have trained enough number of trees. Then we will also need to specify the maximum depth of tree, maximum features per split, and minimum samples per split. So these three parameters is usually trying to control the problem of overfitting. So when we are develop or implement our classification tool, we need to optimize these parameters. And I'll talk more about how do we do the validation and optimization in the following slide. So here is just the process, the development phase, how we develop our classification tool for neuron in Avia. So we use a random forest as our core algorithm for this development. Then thanks to the sample with label for different type of neuron that are provided by neuromorpho.org. So we utilize these label samples to develop our Pareto classifiers for a generic neuron types according to their morphological feature. Then in order to validate the performance of our classifier, we also use k-cross validation to check if it is overfitting and try to verify its accuracy. So here I'm going to talk about what's k-cross validation. It's actually also a very important process when we want to validate the performance of any trained model. So,
so is this what we'd use to make sure the variance is within control?
Yeah, and also try to check it is not overfitting.
Okay.
So overfitting is a problem very common in machine learning model. It happens when we observe the performance in our training data is very good, but when we apply the model being trained by this training data to different test data, sometimes we will observe that the performance is not that good. So the reason is that our trained model its parameter or its structure is overfitted to training data we use. So in order to test whether our trained model is overfitting or not, we will need to use different test data to get a fair evaluation of our model. So K4 cross-validation is based on this idea. So it's actually, if we have this is our entire data, then K4 cross-validation will divide it into K folds, and each fold is independent to each other and with equal size. So for example, if we set K as 4, then we will have data divided into 4 folds. Then we will do the iteration of training and testing with the number of iteration equal to k. So in this example, the k will do 4 iterations, and in each iteration we'll use one fold of data we previously divided as the test data, and the remaining data is training data, and usually training data has larger size than test data. Then we repeat the training and testing 4 times, but every time we are using actually different training and test data. Then in this way we'll get the accuracy from this k iteration, which is more statistical meaningful and also more realistic to evaluate our model. So if we try to check if our model is whether it is overfitting or not, we need to see if the mean accuracy is good enough, and if the standard deviation is small enough. So that's the very important process when we are practicing random forest or any other machine learning model. Okay, so another good another good use of K4 classification is actually can serve as a benchmark for us to fine-tune the parameters, just those parameters we mentioned, like maximum number of depths, minimum number of sample per split. And we use the K4 cross-validation to optimize our parameter for our classifier. And so this is the Pareto classifier we we have in Avia 5, we have human with 4 types of neuron, and with mouse that's for 6 types of neuron, and rat also 6 types. And these are the accuracy we got from the cross validation. So overall, they are working reasonably well with the accuracy always greater than 80%.
Mm-hmm. So I wonder if somebody wanted to look only at, let's say, tripolar neurons, but they had different subtypes. Could they create their own classifier in Avia to do that? So look at just the tripolar and then retrain using just that data.
Yeah, absolutely. We are just picking very generic type, but I think there are still some interesting feature to dig out for some specific subtype. Like the type we pick are very generic, but it may be possible that motor neuron at different location of the brain may have its morphological feature difference.
Yeah.
So I think that's a very good use of our tool for the customer to further define their own type and then train the classifier.
Yeah, and you don't. And how much data here? You have a few numbers.
Yeah.
So the second column after the name is the number of cells that was used for that particular class.
Yeah.
And what's a good, rule of thumb number?
So in general, numbers. More is better. That's a very rule of thumb in machine learning. But I mean, in our data, some of data is maxed out on neuromorpho. So we see that some like granular only have 5, or stellate only has 7 for human. But at least the total number of data should be at least, I think, 50 or over 60 to have its statistical validity. So the more is better, but if we could not get so many, at least we should have 50 or 60.
Yeah,
that's the number we should have.
Okay.
Yeah. So, and Luciano is going to show some demo for our classification tool on Avia 5. So I'll let him show the feature.
Thank you very much.
Yeah,
thank you very much. So Tze-Yuan will stay with us and answer questions at the end. So right after the demo, we'll have a question and answer session. And so I'll go into the demo. I'll come a little bit closer here so I can reach the keyboard if I need it. So we've covered a lot of the details, and by now, if you're a biologist, you've never used machine learning, you might be thinking, wow, this is really complicated. I have no chance to ever, master this. Now, the good thing is we have made it as easy as possible. So if I go into the software, you won't need to do or calculate any of the things that Tze-Yuan has mentioned, because we've already done it. We've already built the whole infrastructure here. So let me get here. Okay. So this is Avia 5, and if you want to download a trial version, you can on our website. It's drvtechnologies.com. You can go there and download it. It's the big button on the top right, and try it out. And as Tze-Yuan mentioned, we've used the established database for published neuron reconstructions called Neuromorpho, and we thank them for allowing us to use the data to create these models, which are open source, so anybody can have the models we have created. And these models we have loaded here. There's 3 of them. Tze-Yuan mentioned the human, mouse, and rat. And we can apply those models that are already validated, they are pre-taught, and you can just hit the button apply, and it will apply the classifier that we've generated on any new data set that you load into the software. So you do need to load a 3D reconstruction of a neuron like this, and I'll give you a little bit more details in a moment. So for those of you who are not into the informatics side of things, and you just want to use a machine learning tool that works, this is what we have tried to do here. So you'd open up the software, you would load your image with the 3D reconstructions, which you can create in Avia or you can create in other packages that are available. You can load them in. We've created the compatibility so you can load in reconstructions created in other packages, and you would have an image like this. So you have the reconstructions on the left here. It shows you all the neurons that have been detected on the right. And down here it shows the classes that we have pre-taught. And I know that this dataset is of human neurons, so I'm going to choose the human neuron classifier. And all I have to do now is, and you can see here the neurons themselves, the ones that I have detected. So there's one there. There's another one here. I'm choosing the wrong one. So here, so this will select the whole. So that's one neuron. Here's another neuron, and so on. So those are the neurons that we have detected. They are also shown here as thumbnails, and you have the 3D view. And what I will do is simply apply the classifier. And right now, there's nothing here. I'm just pointing out there is nothing next to all objects. There's nothing. Once I apply this classifier, there will be multiple tabs at the bottom, which will show in each tab the neurons that belong to that class. And also we can color code the neurons based on class. So I'll do that now. And the time it actually takes to apply is very short, is a few seconds, maybe 1 or 2 seconds. So I'll go ahead and hit apply. And the classification is now done. You can go into this class. So granule, we found 1, 2, 3, 4, 5 of this type. There's one right there. There's another one, and so on. If I go to pyramidal, there's quite a few more. So that's one right there. There's another one. There's another one. They're all kind of clustered around here. And if I go to spindle, there are none, and stellate, there's quite a few as well. So that shows that the classification has worked, and we can inspect the actual cells that have been classified to make sure they are of the correct type. And to help us, we can also do this, just change the color coding based on class, and now we can see them all separate. So this is one class over here, another class in the middle, and another class over here. So this shows how to apply a classifier. Now, if I wanted to build my own classifier, I can just open the classifier view, start new classifier, so create new classifier, and I can choose if I want to use the whole neuron structure, if I want to use information related to the soma, dendrite, or spines. Now, today I'm going to illustrate this just with dendrite, and this will create a new gallery with 82 dendrite branches. And each branch comes from a different, could come from a different neuron. And here in this kind of example, I'm no longer interested in the classes or the types of neurons, but I want to classify dendrite branches based on something that I see. So I'm going to go ahead and pick objects that look to me similar, and I'm going to create some classes. So this is a dendrite that only has one branch. Here is another one, here is another one, here is another, and another. And I want to create a class for these guys, and I assign them there. So there's 1, 2, 3, 4, 5. Maybe I should add a few more. Assign. So there's a few already. And then I'm going to create another class, and I can change this. Single. This is basically single branch. What I'm it's my hypothetical class, and I'm going to create another one with multiple branch. Okay, and I'll look at the main gallery again and pick a few that have multiple branches. And this and that. Okay, and I can assign it to multiple branch, and that's what it looks like. So that I haven't taught it yet. I just added examples, and I could have added also directly from the view, so I can select directly in the 3D view and it will select over here. But today I just do the demo showing that I can select from the top here. I put them in the right folder, and now I'm going to teach. And when I hit the button teach, this taught will update to the number of objects that I used to teach. And also the statistics will be calculated. So the confidence levels will be calculated. The teaching is now taking place. So it takes maybe 1 or 2 seconds as well. And it gives us a confidence level of 92% for the whole classifier. And then per class, for this class, it says 100%. And for the other class, 87%. So this is relatively low, 87%. But overall, this is, as an example, you can see what the workflow is. Once I do that, I can now apply this new classifier. I can save it also if I want to share it with somebody. But I can apply this classifier to the image, and now I have it applied. As a single, I have all of these dendrite branches that are listed here. And then multiple branches, I have all of these that are listed here. And they are also color-coded. You can see all of the items that are multi-branched are now color-coded together, and the ones that are have few branches or only one branch are color-coded another color. Now, this all seems kind of trivial, but the work that took place in the background is quite impressive, because all of your parameters and all of the features calculated from those parameters were used to create this classification. It's not just one parameter. We didn't look at numbers of branches and applied it. And that's why sometimes you see objects that have more branches, but overall they have some similarity, an overall similarity, which might be to do with the tortuosity or a mixture of parameters. And that's where the power of this comes from. You create a classifier that is built on top of hundreds or even thousands of features. And so the confidence level is very high, and it's very robust to noise in the data. So that's a brief demo of how to apply a pre-existing classifier that we have created, and how to create your own classifier. And I already mentioned you can create classifiers based on any of the objects that you detect. I showed today dendrites, but I could have done it also for the whole neuron or for spines, for example. So I think that's it I wanted to show in Avia itself. I'll go back here to the presentation. Okay, so thank you for staying this long. I appreciate it. We will have the questions in a moment. I just wanted to mention that you can bring in reconstructions that you've created in NeuroLucida or in Imaris or in other applications. So we support SWC, IV files, WRL files, ASC files. We can read all of those into Avia, and of course we can read our own that we generate. We can read them and bring them into Avia. You can then apply an existing classifier. You can augment, meaning you can load the human classifier that we generated, and you can add more examples to make it more relevant for your data. Or you can create your classifier from scratch. And then you can apply that classifier on the whole data set within 1 or 2 seconds, and then continue to explore the results either with our plots or by exporting the results into another package to explore it. In terms of accessibility, we have a few ways. You could be part of our DRV beta program, which is also on our website. Currently it's full, but if more openings come up, you can register your interest, and it will be in order of showing the interest. So there'll be a queue, a waiting queue, and yeah, we will contact you if any openings come up. You can also purchase Avia and use it. You can download the trial version from our website anytime. It's right there now. It's version 5.02, and very soon we'll have version 5.1, which comes with some additional features, and we'll have a webinar about that in the near future. We also welcome partnerships, both academic and with industry partners in pharma and biotech. We have a lot more machine learning tools than the ones we've described today. We're also involved heavily in deep learning approaches, and we have a few interesting use cases to share with potential partners, and we're interested in new partnerships all the time. And finally, we can also develop custom implementations and custom validation for your project or for your product. And that's something that we're very familiar with and very comfortable doing. So all of the tools that I mentioned today, plus many more that you can find on our website, are available to you either with the off-the-shelf software, which is Avia, or with any of the other options. So in conclusion, manual selection of key features and parameters can be really time-consuming and very error-prone because you're narrowed down to a couple of parameters often. And because you're choosing the 2 or 3 parameters that you think are most important, that introduces an enormous bias into the analysis, and of course underuses the data set heavily. If you use supervised machine learning to do the same thing, you're straight away using the full depth of the data. Nothing is wasted. And you can apply the classifier that you build with minimum input, a very short amount of time you can create a classifier, to an unlimited number of data sets. So you don't need to go through every single one. You can just apply, and you get a great result on every image. Every image will have all the segmented objects classified accordingly. And of course, it is not as biased, of course, at least not at the feature level. There's always bias, and we can always argue that there is bias in the model, but at least at the feature level, there is no bias introduced by the user, because through the methodology that Tze-Yuan explained using random forest, you're doing a random sample of both the data points and the features within those data points to create this forest of random trees. And then through the voting system that we looked at, you then create a decision made for you. And with that, I want to thank everybody that attended this webinar. Thank you, Tze-Yuan, for your presentation, and everybody that helped us organize the webinar. Willie, Queen, and Hoyen, thank you very much. And we are happy to take any questions that came in the meantime. And you can post questions now still, either on Twitter, LinkedIn, or on our website.
You did too good of a job. There were no questions.
Okay, good. So I think we have covered everything for today. If you have any other questions, you can email me directly as well. My email is luciano.l@drvtechnologies.com. That's luciano.l@drvtechnologies.com. And I'll be happy to follow up with you. Thank you very much, and I wish you a great afternoon. Bye-bye.
Bye.